You're welcome, Prof.
All protocols duly observed. Mr. Governor, Your Excellency Governor, and the Commission of Health, Lagos State, it is a pleasure to be here and stand in front of you. I call you my heroes because what you did for COVID and your response, for Lagos, Nigeria, and Africa is unparalleled. Thank you very much for the work you've done. So today, I want to believe that the commissioner gave me an assignment, and that should be to talk about the gaps, how do we bridge the gaps of genomics and bioinformatics to address the issues of public health and especially epidemics. So, I represent this laboratory. Yeah. So, and the issue of genomics is paramount simply because a few years back, we realized that genomics was a science that really changing the paradigm in medicine and research, but Africa wasn't contributing to it simply because there was no knowledge on the ground. And that created a huge divide between the rest of the world and Africa. And what we did was basically to think about how do we bridge the gap. And you could see this diagram describing our approach to bridging that gap: education, infrastructure, and partnerships, translation, and research. And then, application, and especially in infectious disease, because that's one of the major things that plague Africa. And we set up a platform, and that platform is called the African Center of Excellence for Genomic of Infectious Disease. And it is a conglomerate or consortium of academic and clinical testing centers in West Africa, and also with partners overseas. And the mission of that platform is actually to bring Africans together so that we can actually use knowledge of genomics to address problems of infectious disease on the continent. And we used a two-pronged approach for that. One was education, and then the second one was research. And then the research was focusing on febrile illness. The reason why we chose febrile illness is simply because many of the diseases that plague Africa present as fevers. But within the fevers, there are different types of fevers. And as we see in our own hospitals in Nigeria, the first culprit is often malaria. Then if it's not malaria, then you think about typhoid. Doctors are very, very creative. These days, because they don't want you to come back several times, when you come, they say, well, you have typhoid malaria. So they bombard you with both antibiotics and antimalarials. So, but then the idea is in the program to identify the drivers of febrile illness, and understand what we call the microbial landscape. And once you understand the microbial landscape, you can actually develop the right countermeasures. And those countermeasures could be vaccine, diagnostics, and therapeutics. And that's what we did. In doing that, we meet one of the major needs of Africa, which is, building that critical mass that Africa needs for research and bioinformatics. But ultimately, the goal is actually to build the next generation of African pathogen hunters, hunting these pathogens in Africa, in the field of the continent, and then start playing what I call offense. That is, understanding them, characterizing them, and developing the countermeasures before they come to us. And we actually made that first demonstration during the Ebola outbreak. In the first week of the Ebola outbreak, I remember Professor Gebao coming to my lab here in Nigeria when we were still thinking about Ebola spreading out of Guinea, and we set up a strategy. And that strategy was set up diagnostics. And we detected the first case of Ebola in Sierra Leone, just like we did for Nigeria. But what was important is the fact that we deploy genomics in the field during an outbreak for the first time in the world. And we sequenced the first 99 viruses in West Africa, made them openly accessible, and then shared it to the world so that the world could use it for diagnostics, therapeutics, and vaccine. So in real sense, we pioneered open data sharing and open data access. What you see in GISET today is something that we started here in West Africa in 2014. And we accelerate the process of sequencing, creating new ways of doing these technologies, from sample collection to sequencing the whole genome and depositing this data in GenBank in 10 days. This was a feat that was never achieved. But we identified mutations in the virus, and for the first time also during that outbreak, we were able to characterize mutations that give the virus the ability to spread faster. And that's why we hear mutations that are associated with increased transmissibility. That was done in 2014. We're happy that that is a trend. But we use genomics as a way, as you see there on the diagram, on the chart. It's not working. But apart from that, we really translated genomics into something, into point of care. That is, going from genomic sequences, we developed the first rapid diagnostic test that was approved by WHO and then the US FDA under emergency use authorization, cutting down the diagnostic time from hours to 10 minutes. And that really was the first time where we could show that in Africa we could rise to the occasion and respond. But then I will move fast forward and then respond to something that is very important, especially for Nigeria, Lassa fever. This is a disease that was described for the first time in Nigeria about 53 years ago. But then that disease up to 2008 did not have molecular diagnostics. We worked with colleagues in Nigeria and in Ebola Specialty Teaching Hospital, setting up the first molecular diagnostics. But what we did also that is incredible and that has changed the dynamic is the fact that we generated the first genetic sequence of the virus. And since the time we generated the first genetic sequence, there are three vaccines in clinical trials. So showing the power of genomics in terms of accelerating solutions for problems that plague Africa. We also developed a rapid diagnostic test for a virus of that nature. So we've also gone on, as I mentioned before, discovering 2 new viruses. But then these viruses, we discovered them around Epoma. And we call them Epoma virus 1 and Epoma virus 2. These 2 viruses were very similar to a virus that created an outbreak in the DRC, and that was responsible for a few deaths over there. Now, as we all know, in 2018, Nigeria experienced a huge surge in the number of Lassa fever cases. In 2 months, Nigeria had more cases than the past 2 years combined. There were many unanswered questions. What was driving the surge? What was causing the people death? And we were able to answer that within a week, collecting samples, generating genome sequence data, working with, doing this within country, and really, this is a group of young, talented Nigerians taking responsibility and moving the field forward in a way that was unprecedented. We were able to answer the questions. No evidence of particular new virus, no evidence of sustained human-to-human transmission. This information were given to the Nigeria Center for Disease Control, and then this was used effectively to respond to that outbreak. Here you could see, for instance, the WHO and NCDC acknowledging that effort. But then what is important is the fact that we, again, change the tide. That is moving the technology forward, using genomics to create a new rapid diagnostic test. And this is the first nucleic-based diagnostic test on a paper strip. And this was done for Lassa fever. And you could see here in Nature, where, in the journal Nature, where the journalist embedded in our lab was able to demonstrate this, cutting down the cost of testing from over $100 to about $3 a test. So again, this shows you here, for instance, in our case, where in 2018 also, toward the end of the year, within 2 weeks, 179 Nigerian children dying in a village around Edo State. The cause of the disease was never established. All tests fell negative. Yellow fever, Lassa fever, all of them fell negative, Ebola. But then the question was, what was responsible for the death? NCDC sent the samples in our lab, and within 48 hours, we were able to actually identify the fact that we had a lineage or a strain of the yellow fever that was different from the one that had been circulating in Nigeria for the past 94 years. And that really, we sent the signal back, they declared an outbreak, and within 2 weeks, along with the National Primary Care Development Authority, they were able to contain that outbreak, saving thousands of lives. Again, this is an example showing how you could use technology to respond to an outbreak, just as the commissioner mentioned. We recently just described the very first genome of the extensive drug-resistant tuberculosis in Nigeria. And this is the very first time, a tuberculosis strain that does not respond to any of the antibiotics, and that really should be worrisome. So this information has also been shared by public health authorities, but this is the first time this is described in Africa. We are not just doing the science, we're also developing the tools and the technologies to respond. Here you could see 2 young Nigerians developing bioinformatics pipeline. We are very much aware of the fact that internet is a problem. Here, these are tools that you can use on your computer without internet. Do genetic analysis, generate genome sequence data, and then you can use this information to respond to public health threats. For COVID, I will just move very fast. It was a very incredible collaboration between the Lagos State Ministry of Health, the NCDC, and us. Nigeria set the pace for Africa. Within 48 hours of receiving the samples, we were able to generate the first genome of this virus on the continent. And that really, again, I will say, the High Commissioner, the Commissioner for Health, that was an incredible collaboration, and I think that's the way we should be working to actually put this country, and give this country the glory it deserves. We wanted to work with government, as you see here on the map, generating in real time the number of variants that are circulating in different states in Nigeria. So we could see there that whereby in time and space, we know all of the variants that are circulating in the country. These information are often given to the NCDC so that they can present it to the Presidential Task Force. You can see here, the variants across time. And as you can see, as of September, almost 100% of all variants circulating in Nigeria are Delta. So it shows how transmissible this variant is. So we can see the variants of concern. You can see here the new variant that we described first time in Nigeria. But then what we've also done is actually to develop new technologies that can analyze this variant and then can make prediction to know those ones that will become variants of concern. So that's basically just showing an algorithm there. We also develop a rapid diagnostic test, for risk, as I mentioned before. Now we can combine all of these tests. We have the ability now to combine over 140 different viruses in one single paper strip test. And then you can identify that and get the result in near real time. But we've also built capacity on the continent. We brought together over 14 different African countries. We've generated over 5,000 genomes across the continent. And countries like Rwanda benefited a lot because they send their sample here, we sequence, send it back to them, they declare lockdown, and then they were able to control, outbreak. We're also developing tools, tools that are very important to places like Lagos and other state dashboard, whereby the commissioner or the governor could be here looking at his, on his phone, and then knowing exactly what is happening in each local government or in the nation or across the world. And these are some of those tools here, for instance, where you could see a little demo. You could see, we look at the delta. And if you look at the delta, it gives you what happens in the world, either the gamma lineage or variant. And you have that image in real time. You don't need to go and look for, on the internet. You just have an app, and it gives you this information in real time. You know what's happening in your country, in your local government, and everywhere in the world, in real time. So with this, again, and then you can also think about looking at the Delta variant, even in the context of just Nigeria. You know, with this, you can know exactly what is happening, and you can compare it with others. And with this, you don't have to be a genomic, you don't have to be literate. Just look at it as you are seeing here now. You could look at it and then know what are the different mutations on the genes. So really bringing it to a level of people, for people that are making policies. You don't have to be in the someone in the lab to do all of this work. And that really has been very helpful. So this diagram gives you an idea of what you could see. I took the case of Lagos. This is I'm developing in our lab. And then just looking at the Delta variant, within 24 hours, Mr. Governor could be sitting here and then know exactly what was it yesterday, what it is now, without having to call on somebody to share data. And then this is really bringing data to the fore, and then making people making decisions that are informed based on information. Not having to have meetings of people, but actually having the data that are uploaded, and then you can make informed decisions. So I will close in by just talking about this new program, and it's called Sentinel. It is a pandemic preemption and response system. The Honorable Commissioner alluded to that. How do we look into the future? We need to have the ability to start preempting pandemics and then using technology to preempt it. And it's built on three major pillars that I mentioned there. But what is important is actually to create an ecosystem. And that ecosystem is based on what I call the hubs and spokes approach. Lagos is building, a very fantastic laboratory, as just the commissioner mentioned. That could be built, that could become a hub, for this kind of system. And then these hubs are connected to the spokes. And then the spokes could be actually the peripheral institutions. And then with this, we can now empower the workforce in a way that we can all work together and then become a community where everybody is looking after one another. Ultimately, our goal is basically to create a platform where we can interact with partners, bring technology down to a level where we can actually use in order to respond and help Africa to be in the leadership. In closing, I've always said before, God was not stupid to create Africa. Africa is rich not only in mineral resources. God made Africa rich in its own biodiversity. And as part of the African biodiversity are viruses and pathogens. Africa should be the leader in the world in providing solutions, but also in showing the world how to do things. And I think looking forward, we should claim our place, which is these viruses are ours, and let's take it and make a change in the world. Thank you.